Okay, so let me start my. All right, so thank you everybody for joining today. As you know, we all love talking about our favorite topic, and my favorite topic is climate change microbiology. And I'll be discussing methanotrophs today for mitigation and removal of methane. So part of what I do also is look at the intersection between the methane cycle and the nitrogen cycle. I'll touch on that a little bit, but if you have any questions about how nitrogen is involved in controlling the methane cycle, I'm also happy to discuss that with you at a later point. So there we go. So let me just start. I'm not going to give we all know climate change is happening. We all know that methane is one of the major greenhouse gases. And we also, in this group, likely understand that the mitigation of methane is likely to give us the best short-term option for limiting the rate of temperature increase, because methane is a shorter-lived gas in the atmosphere than CO2 or nitrous oxide. And removing methane could limit temperature increase on a decadal scale rather than a 100-year scale. So focusing on the methane cycle, we can start pinpointing the important sources of methane and then the sinks of methane that could help us mitigate it. So what's been noticed over the last, I guess, decade or so is that the majority of methane that's entering the atmosphere right now is biogenic. And so this is the updated global methane budget from the Global Carbon Project. And what this shows in the orange boxes is the contributions of sources of methane by methanogens. And these are archaeal microbes that live in the environment. And when there is plenty of organic carbon and anoxia, they love to pump out the methane. So from agricultural and waste and a large substantial source from combined wetland and inland freshwaters, this is where methanogens are really thriving. And they also contribute a bit to these two sources and other emissions, but really the big sources are in the agricultural and waste sector and then in the wetlands sector. Now, if we look at the biological sink for methane, it's not nearly as dramatic as the atmospheric sink. So the largest sink for methane is hydroxyl or chloride atoms in the atmosphere. And there is other work that's going on in trying to augment the oxidative capacity of the atmosphere to control or remove methane. But we're focused on the biological sink, which is the methanotrophs. Now, even though this is a fairly small sink piece, we also have to recognize that methanotrophs are controlling methane emissions from the environment even before it enters the atmosphere. So the biological methane sink is actually quite potent and powerful. And our job in learning how to manipulate them is trying to figure out if there's a way to increase their sink capacity. Can we use them to bring more methane out of the atmosphere or control methane before it's emitted to the atmosphere? So where do we find methanotrophs? Well, they're within soils. So wherever there's a methane source, we tend to find methanotrophs that accompany them. That means that there's also methanotrophs that have been found in the digestive tracts of animals. There's methanotrophs that have been found in landfill soils, in wetland soils. But the ones that have been characterized the most are from soil ecosystems or in aquatic ecosystems. And this is an image from the Encyclopedia Britannica. I've modified it a bit because we know that, for example, in aquatic systems, it's not just anaerobic processes that remove methane. There's also aerobic methanotrophs that use different electron acceptors than oxygen that are also active in these ecosystems. And that's really important for us to understand, because even in ecosystems without oxygen, we can still think about how to augment the aerobic methanotrophs in those ecosystems. So how do we do that augmentation? Where can we augment methanotrophy? And this is an image from the National Academy's report that came out in 2024. And I was a committee member writing that report. And what we were doing here is specifically looking at ways that we could control atmospheric methane. But I'm going to use this image for all biological methane removal possibilities. Well, maybe not all, but the ones that we've envisioned thus far. And I guess our challenge as a community is to see if there are other more creative or unknown ways that we can augment methanotrophy in ecosystems to either remove methane or prevent its production. So the 4 areas that I'll be focusing on in the next several minutes is these 4 areas. So we can engineer trees. Trees can be a big, they're obviously a large biotic force of nature. And if we can augment the natural methanotrophic component of tree microbiomes, then we can use them as uptake systems. We can also look broadly at soil amendments. We can look at aquatic methane removal, and we can look at methanotroph bioreactors. So these are the 4 that I know the most about. I'm hoping to learn more about other types of ways that we can use methanotrophs in our industrial sphere. So I'm going to start with methanotroph bioreactors, which is the space I know the most about. And this is where my lab has been doing active work. So first of all, just a brief introduction to methanotrophy for those of you that aren't so familiar with the biological side of methane removal. This is using microorganisms called methanotrophs that consume methane as a sole source of energy and reductant. And the way that they do this primarily is that they have a membrane-bound monooxygenase enzyme that's loaded into these membrane layers that form within the bacterial cell. Now, there's also a cytoplasmic methane monooxygenase. That one is not quite as abundant in the methanotrophs that we know about. It does play a role in methane removal, and people are looking at modifying soluble methane monooxygenase. But for the most part, we think about the membrane-bound monooxygenase just because of its vast abundance and its capacity to allow these microbes to remove methane. So these are really cool bacterial cells. You can see in this electron photograph that there are these layers of intracytoplasmic membranes. And within these layers, if we zoom in, what we can see is that they're infoldings from the cytoplasmic membrane. And the enzymes that oxidize methane are completely packed within these membrane stacks. So if we zoom into the stacked layers, what we can see is in this little circle here is that there are these crystalline protein configurations of the methane monooxygenase and the enzymes that are part of the methane oxidation pathway that are loaded into these membranes. This gives the cell an overall huge capacity to collect methane and oxidize it. Now, when I say huge, there's still a lot of limitations kinetically to methane diffusion to the cell and the rate at which these cells can oxidize methane. So unfortunately for us, it's still too slow at this point just to rely on the natural methane oxidizing capacity of bacteria, but we can work to augment their ability to oxidize methane more efficiently to make a dent in methane removal. So this is just a schematic of the pathway for methane oxidation. So the monooxygenase, denoted by MMO here, oxidizes methane to methanol, and then these intermediates are sequentially oxidized to carbon dioxide. So when we're talking about methane removal by biology, we're really talking about transforming methane to carbon dioxide, which of course is also a greenhouse gas, but it's 80 times less potent in holding in heat on a 20-year time scale. So converting methane to CO2, it has an overall climate beneficial effect. And then if we can focus also on removing CO2, then these 2 pieces of our climate change technology work hand in hand. So removal of methane and CO2 can work to bring the greenhouse gases down. Okay, so in feeding methanotrophs, they are using methane as a sole source of carbon and reductant. And so what that means is that they can derive their cellular carbon and all of the energy that they need for the cell from using methane as their sole energy source. Now, of course, they also need a lot of micronutrients. They need trace metals, they need a nitrogen source, they need a phosphorus source. So those things can be found in media, in water. We add that to the media in the laboratory, but methane is their primary energy and carbon source, and we feed these to bioreactors. So in methane bioconversions technologies, the idea is that we can harvest methane from any type of source. So we can take it from flare stacks. So instead of burning methane, we can pipe that methane into these bioreactors. We could harvest methane from landfills. And then there are companies that are active right now that are converting methane in bioreactors containing methanotrophs into biofuels, into single cell protein, and into biopolymers like plastics. And this is an image from Mango Materials that's in the Bay Area that is doing just that by connecting methane bioreactors to wastewater treatment systems. They feed their methanotrophs the wastewater methane, and they're making fabrics and polymers for their company. So the types of methanotroph bioreactors that can be used, these are the ones that are relying on the ability of the methane and oxygen to reach methanotrophs in a liquid mass. So these are essentially stir tank types of reactors, whether they're bubble lift or airlift or stir tank. There are limitations to this type of bioreactor. And the main ones that have been identified are the mass transfer limitations of the gases to the microbes. The growth rates and kinetic constraints mean that the cells don't grow as rapidly or to as much yield as we would want. So these are limitations that that scientists are working to overcome on lab scales, and then hopefully moving them to scale up. So some of the ways that these limitations can be overcome is by attaching the biomass. So membrane bioreactors attach the biomass to membranes where the cells are fixed and then the gases are passed by them. A fluidized bed is where biofilms of methanotrophs are on carriers that are moved through a liquid interface. And then this is another type of packed bed bioreactor where the biomass is attached in a fixed packed bed, and then the materials are able to diffuse more readily to the microorganisms when they're in this configuration. And this packed bed is similar to what our project is in my lab. And this has been funded by Spark Climate Solutions. And what we're doing is we are using fixed biomass on a microfluidic hydrogel scaffold. So these are highly engineered scaffold materials that are made from a plastic polymer. And each microfluidic chamber can be embedded with hydrogel that has been infiltrated with methanotrophic biomass. So this is the scaffold where the microbes and hydrogel are affixed. And the hydrogels can be PEGTA, which is what our collaborators at Lawrence Livermore National Lab have done. That's how they initially created it. But we can also use less expensive hydrogels like alginate or agar. So the idea here is that because the microbes are within the hydrogel, there's no mass transfer limitation in this configuration. And that means that even for regular methanotrophs, if we are using these scaffolds, they can consume methane down to subatmospheric levels. So these are real measurements that were made by Fang Chen in her lab, showing that these microfluidic bioreactors can consume methane at 1 atmosphere, and then 2 atmospheres is atmospheric concentration. So it's possible, it's just not optimized for like removing atmospheric methane at a high enough rate to make a difference. So our Spark project is to get to that point where we can demonstrate that this is a viable technology to remove atmospheric methane at scale. And our idea is that we're going to compare regular methanotrophs and what we call atmospheric methanotrophs. And atmospheric methanotrophs are these organisms here. This is Methylocapsa gorgona. It still has those membrane stacks that I showed you in the previous slide, and it grows in these nice biofilm configurations. And these microbes were isolated from an Arctic pulsar and have been shown to grow on floating filters on air alone. So they are true atmospheric methane oxidizers. And this is a comparison of the growth on air by a variety of different methane oxidizers. Here's the Methylocapsa organism. This is the amount of gases that they're consuming over time. You can see this is a pretty long time frame and not a very high concentration because this is air that they're consuming. So this microbe can consume methane, carbon monoxide, and hydrogen at the same time. And for us, that's fairly exciting because it means we can get a mixed gas removal using these microbes. Now, it's not the only one that can do this. This is a comparison with other types of methanotrophs. And you see here this Methyllocystis rosea, which is a typical alpha proteobacteria methanotroph that's in culture collections. It also has the capacity to grow on air. So it's not a special feature of this Methylocapsa gorgona type to grow on air. And that's also exciting because it means that many of our methanotrophs in culture are likely capable of this activity, which gives us more capacity to use different microbes to scale this process for atmospheric methane removal. Now, why might we want to pursue atmospheric removal? That was the big question that the national academies had to answer. And the answer is still actually up in the air. I don't know if it's important to pursue atmospheric methane removal given the costs and limitations of it. But one reason that we might want to pursue it is because as we replace fossil fuel energy sources with hydrogen, there is modeling data that's out there that suggests that hydrogen leaking from production facilities could increase the lifetime of methane in the atmosphere by decades. And the reason is that hydrogen will compete for the hydroxyl in the atmosphere, which means it won't be reacting with methane. So if hydrogen is removing that oxidative capacity, then methane has nothing to react with, and it will persist. And of course, it has a very high global warming potential. So that's one reason that we might want to consider atmospheric removal. So how can methanotroph bioreactors make a difference? Well, in a paper in 2023 that was published by he et al. From Mary Lidstrom's group in the Proceedings of the National Academies of Sciences, they had a wonderful study where they were showing that in bioreactor systems that can consume atmospheric methane concentrations, if we could deploy 50,000 to 300,000 bioreactors, that can consume 40 megatons to 1.7 gigatons of methane over 20 years, and this is a possibility, then that would make a difference to the rate of temperature increase. So this is a reasonable target. Like in the biological sphere, this seems achievable. For the methanotrophs that were used in this study, they worked the best at emission sources over 500 ppm. Atmospheric is 2 ppm, but this gives us, again, some play space for between mitigation of methane and atmospheric removal. And it doesn't have to be either or. We can use methanotrophs for both. And then, as mentioned, with higher methane emission sources, we do have the possibility to recover single cell protein, which is the microbial biomass itself, or we can use methanotrophs as machines, which is shown in this image here, to create different types of bioproducts. And so this is also something we pursue in my lab, is understanding the range of bioproducts that methanotrophs can make. And at high emission sources, we can get to biofuels or biopolymers or other types of materials. And then because methanotrophs can consume hydrogen at the same time, at least the atmospheric, under atmospheric conditions, it's possible that we could use methanotrophs to co-remove hydrogen and methane so that when hydrogen leaks from those production facilities, maybe methanotrophs could also be used to mitigate that hydrogen and methane. So that's something else that we can think about. All right, so moving on to soil amendments. So soil amendments are also being pursued in a number of labs as a way to mitigate and remove methane. So this is just showing that as temperature increases, the relationship between plants and the microbiota creates this priming effect for carbon cycling. And as we get higher temperatures, that priming effect is stronger, which is modeled to cause release of increased methane and CO2. So it's really important to start thinking about how we might work to control greenhouse gas emissions from soils as temperature increases. Now, well-aerated soils are usually net methane sinks. They're not sources. And that's because, as I mentioned in the very beginning, methanogenesis occurs in anoxic soils with high carbon. In well-drained soils, this is mostly well-aerated and has less soil organic matter. So the methanotrophs do a pretty good job at filtering the methane. However, if we look at flooded soils, which is our best example is rice paddies, then these soils become methane emitting sources. And that emission is from ebullition, from just the vast amount of methanogenesis that occurs under these flooded conditions, and also just the methanogenesis overwhelming methanotrophy. So we all know that rice paddies are something that we have to think about mitigating. Now, one of the ways to mitigate methane emissions from rice paddies is to drain the rice paddies. And you can still cultivate rice without flooding them. However, what's been noted is that because rice is still fertilized with nitrogen sources, when rice paddies are drained, they tend to turn into net nitrous oxide sources. And nitrous oxide is 300 times more potent than carbon dioxide, so way more potent at small concentrations than methane or CO2. So we do have to keep mindful that if we are mitigating methane, we might end up inadvertently increasing nitrous oxide. So that's an issue that's shown in this feature here. Now, this was on this side of the slide. This is from a study that was showing what happens between submerged paddy soils and drained paddy soils when there is a carbon fertilizer addition versus nitrogen versus a carbon plus nitrogen addition, and what happens with nitrous oxide emissions. So when the paddy soils are submerged, nitrous oxide is not emitted. However, when they're drained, then where there's nitrogen, that's when nitrous oxide is a problem. So the way to amend the soil is to find a way to control the nitrous oxide once the paddies are drained. And so this is a study from 2018 where they did just that. And what they were doing was looking at two different types of treatment. They added a mulching treatment, which is a bona fide soil amendment. And then they also looked to see what would happen if they mulched the soil and added a nitrification inhibitor, which is called CP in this slide. And so what they found is what you might expect, that if they have flooded soils, then they have high methane, but not very much nitrous oxide. If they mulch, then they tend to get low methane, but nitrous oxide is still a problem. If they mulched and added a nitrification inhibitor, then they have high productivity, high yield, and they can keep the greenhouse gases low. So this is just one example for how you can manage a soil system, keeping mindful of the types of nutrients that will promote greenhouse gas production because of how microorganisms respond to irrigation, to redox potential, and to available nutrients. So what's happening in the methane space is that a variety of soil amendments are being looked at. And so far, it seems that amendments like biochar or rock dust have been quite effective at preventing methane emissions. And it seems from preliminary studies that these two types of amendments might also be able to keep nitrous oxide levels lower. That's still uncertain because it's all dependent on how fertilization occurs in these soils. Now, the reason that biochar and rock dust seem to augment methanotrophy is because these two amendments provide access to micronutrients, mainly trace metals like copper and iron. And so these become unlimiting to methanotrophs and will stimulate their methane consumption activities. These two types of amendments also tend to retain the moisture in the soil and keep that moisture at an optimal level so that methanotrophy is encouraged to proceed. And then lastly, when these amendments are applied, it keeps the biogeochemical cycling of nitrogen, copper, phosphorus, and other trace nutrients within the soil column instead of blowing out, in dust or whatever. It's not lost. So we all know that topsoil loss is a major problem in agriculture. Adding these amendments tends to retain those soil nutrients and soil quality. So this keeps methane emissions down. Now that it's early days, we don't have like large demonstrations of how this works, but work by Wendy Silver and Jeremy Semrau and others are showing positive results that these types of amendments really could make a difference in agriculture for keeping not just methane from leaving soils, but even allowing for atmospheric methane consumption to occur. Okay, so now moving on to tree engineering. This is the one that I know the least about, but I think it's also among the most nascent of areas for controlling methane with biology. So this is an image from a review article that just covers why trees are part of the methane cycle. It's really not obvious that they should be, but studies have shown that when you measure methane around trees, what you find is that they tend to be similar to rice plants, where they can draw methane up from the soil and release it through their bark. So the closer the measurement is to the soil tree interface, the more methane is measured. And as you go higher up into the tree canopy, then methane can even be consumed. So there's a gradient of how methane is released or consumed in trees so far from what's been measured. Now, there's been a couple, not many, but a couple of metagenome studies looking at microbial communities that reside inside tree bark. And so what this has shown is that there are methanotrophs inside the bark of trees that can oxidize methane before it's released. However, on leaf surfaces, methanotrophs are rarely, if ever, present. However, methylotrophs, which are bacteria that oxidize methanol, are highly abundant on leaves. In fact, that's a fun science experiment to do with kids where you can take a leaf from a deciduous tree, like a maple tree, and put it onto methylotroph growth media, and then the bacteria will grow in the imprint of that leaf. So methylotrophs can be highly abundant on leaf surfaces. And it seems that they might be playing an indirect role in methane cycling. But again, this is nascent ideas at this point, and it's still unknown how methane cycling can work on leaves. But in terms of engineering trees for methane removal, there's a couple ideas that I've seen that have been funded and are out there. One of them is to express a microbial methane monooxygenase in plant leaf tissues. And if you can express a small amount of this enzyme, it won't, you have to express a small enough amount that it doesn't interfere with plant physiology. But if it can be activated, then perhaps those leaves can oxidize atmospheric methane and be used as an atmospheric methane uptake mechanism. And then another idea is to manipulate the microbiomes in trees. So either the bark microbiome and or the root microbiomes. And if you can figure out what's limiting methanotrophy in those microbiomes, perhaps you can add that missing ingredient to stimulate tree growth, allow them to consume more methane and sequester carbon. So this is the idea of manipulating the microbiomes within the trees to try to activate them to be better carbon sequesterers and to consume methane. Now, two projects that have been funded by Spark in this area is with Vincent Gauci, who had a Nature publication looking at one of those bark microbiomes. I think it was last year that that paper came out. So his project is testing the limitations of methanotrophy on tree stems. And then Eli Hornstein is doing a project to genetically engineer plants for atmospheric methane removal. So these are two projects to keep your eye out for, and we'll see what they find in the next couple of years. Okay, so now moving to removal of aquatic methane. So this is really important and is becoming recognized as a much more important way to mitigate methane using microbiology. And that's because the 2024 global methane budget has indicated that the inland, freshwater, and wetlands account for 40% of global methane emissions and is likely increasing as temperature increases. So this is quite a remarkable number. So it means that if we aren't focused on mitigating methane from freshwater and wetlands, we might lose that opportunity. It's a really big source. So combined emissions is 248 teragrams of methane per year. So this is a lot of methane that we need to think about managing. However, you can see from these maps where we have high emissions, a lot of these emissions are in regions like in South America or in Africa or in the polar north, where it's really hard to get to. And these are vast landscapes. So when we're thinking about what to do technologically, we have to consider realistically managed versus unmanaged sources. So one of the low-hanging fruit is to think about hydroelectric dams, so inland waters that are dammed basins that are high methane emitters. And this is what a group or a company called the Floating Island International is doing. And I know that they're here listening to this talk, which is great because maybe they can answer questions afterwards. So Floating Island International has a project where they are creating floating island systems that can remove aquatic methane. And so the reason that you would want to do this in dammed reservoirs is because when nutrients flow into a reservoir, then they tend to pile up near the dam benthic area interface. So we again have this situation where methanogenesis is going to take off gangbusters because now we have an anoxic ecosystem. This is the oxycline. So this is an anoxic space with high nutrients. So we have a lot of methane and it becomes hypersaturated. That means when the water is released from the reservoir, it's immediately aerosolized and becomes a major source. And there's been some popular news articles about the source of methane in the BBC and other places. So what Floating Island International is doing is they have a demonstration called removal of aquatic methane. And they've shown that in their demonstration systems that methane can be remediated from these reservoirs in a year's time. So they're highly effective. And right now there's two steps to their system. And then I'll talk about a third idea that can be included as well. So the first is that aerating the water column is key because this will promote methane oxidation over methanogenesis. It also prevents methane saturated water from leaving the dam outflow. And what's been noticed is that when you aerate the water column, then fish will return, there's less eutrophication effects, water is clarified, and you get speeding of nutrient removal. So you can really increase water quality and remove methane simultaneously. Now that's part of it. And then this is just an image of how if you oxygenate this layer of water, then when it leaves the dam, then there's no issue or limited issue with methane being released. But in addition to aerating the water column, the floating islands component is that this company has made these floating islands that will sit on top of the reservoir system. And it's a biohaven matrix that they've developed that's embedded with aquatic plants. And at the bottom of this floating island is this really thick periphyton. So this is a real image of the periphyton that's at the bottom of these islands. Now, it looks like a bunch of microbial gook, which we love as microbiologists. But what's been noted is that this periphyton is highly enriched in methanotrophic bacteria. So those microbes are actively consuming the methane in the water column. And there's also been some indication that they can degrade microplastics as well. There's only been a couple of studies on that, but I think it's kind of an exciting area to look at too. So also, depending on the type of plants that are put onto these floating islands, it might also be possible to remove atmospheric methane, and that's something that's yet to be measured. Now, again, another issue, since these are eutrophied systems and there's a lot of nitrogen that's going into these reservoirs, we have to be mindful of the nitrous oxide source that's also coming, but we could potentially use these floating islands to co-control nitrous oxide and methane. So again, that's an exciting direction that these can go. Now, the third piece that hasn't been explored to the same extent is to add an amendment to the benthic layer of reservoirs. So it's been shown that zeolite minerals are very good at capturing methane. And this is an image from a group at the University of Basel that's using zeolite cages to capture methane in the benthic or the hypolimnion of lakes, but they're capturing enough methane that they would like to divert this methane as an energy source to burn as fuel. So they think that, so they're developing this as essentially using benthic methane as a natural gas fuel source. For other reservoirs, perhaps we can use zeolite cages just as a way to capture methanotrophs and have them convert methane to CO2 as a way to further clean methane from reservoir systems. But it's a neat idea to use this type of mineral amendment to control methane. Okay, so just to wrap up, the big question is, how much can methanotrophs help with reducing global methane? Now, first I'll start with the negative side, and this is an image from a review that Eric Davidson and Jeremy Semrau wrote with co-authors. And what they are showing here is that the kinetic constraints of methanotrophs are substantial. So at 2 ppm methane, we might have high affinity methanotrophs, but we also have a very low Vmax. And then here for the lower methane affinity methanotrophs, they have a higher Vmax, but can they bind methane at 2 ppm? Possibly, if we can create bioreactors or ecosystems that allow them to do that. But the kinetic constraints mean that we have slow growth. They just don't operate very quickly, which means that our amount of biomass is also limited. So we need to think about ways to grow biomass and then have that biomass work for methane removal, or simultaneously have them grow and remove methane. It's a tricky piece that we have to consider to make methanotroph technologies worthwhile for removing enough methane to have an impact. Another concept is that you could introduce methanotrophs, and I think this is something that Windfall Bio is promoting. The problem with introducing methanotrophs to an ecosystem is that they have to compete with microbes that are already adapted to that ecosystem. Now, just my own personal point of view, having worked with methanotrophs for decades, they're finicky, they're difficult, they're not the easiest microbes. We have enough problem having them compete in our lab space. Now, putting them out in nature, my guess is they would just die. I'm not sure how. I'm interested to know how people can overcome this issue with having introduced microbes compete. And I would love to know if it's possible and why, because I think that would be exciting if you could show that an introduced methanotroph could actually compete for niche space and become that methane sink that we're hoping for. And then another limitation is that ecosystems are vastly heterogeneous. Even bioreactors are heterogeneous if you if you don't have them well controlled, or if you're developing them, then you have to deal with those heterogeneities and what you need to feed them to keep methanotrophs happy and active and growing. So these are all difficulties with biology generally and what we have to overcome. Now, on the positive side, methanotrophs are found in diverse and numerous ecosystems. Where there's methane, there's going to be methanotrophs. And there's a lot of genomic diversity in methanotrophs. So we've been sequencing the genomes of methanotrophs for decades now, and we are just constantly overwhelmed with the amount of metabolic diversity in these wonderful organisms. Some of them can consume other substrates besides methane, which might be another key to working with them and feeding them to have them be better methane sponges. So there's a lot of play space metabolically and biologically with the diversity that has evolved. I'm going to say, although some of you might challenge me on this, I would say the enzymology is fairly well understood. I mean, well enough understood that we can manipulate them. Now, of course, there's always a lot of physiology we don't know about, and there's complexities with how all of the components of methane oxidation fit together and where electrons are going and how carbon flows through systems. But I feel that after studying them for 30 years, that we've gotten somewhere. Like I feel like we have a better handle on methane oxidation to the point that we can do something like this experiment with biochar, where we can dose a field with more and more biochar, and we can see methanotrophs growing, and we can see methane being pulled down. So this is a concept of how biochar is working to bring down methane emissions. So I would say that the early studies on things like biochar and bioreactors are promising. We have enough promising results that I think this is a really important area to invest in. I think that there's a great opportunity. The Floating Island International work has shown great improvement of methane in freshwater systems. So I think that we're on a really good, solid course, and I'm very excited to see what happens with the remainder of my career and young people coming into the field. So with that, I'll give you my acknowledgements and funding sources, and I'm happy to take any questions. And thank you for listening to my bit on methanotrophs.
Oh, amazing. Thank